Okay. Then I first met Steve, we were trying to figure this out somewhere around 5 to 7 years ago at a Gordon conference for microbial population biology. And I haven't seen him since then. But we've communicated via email several times about him coming out and giving a talk, and it's always been impossible to schedule.
Which is pathetic, since I'm an hour down the road.
That's right. That's right. It's not his fault. It is just something. Anyways, but the interesting thing is that when he finally was like, okay, we set a date, and then I hadn't gotten a title yet, and I'm thinking, now he seemed really interesting when I talked to him, but I couldn't exactly remember what we talked about anymore. And then when he gave me his title, I'm thinking, oh, okay, now I remember. It's this nice mix of microbiology, ecology, and I always really love things, especially with spatial structure. So I'm going, okay, now I remember why I invited him. So I can tell you that he's really fun to talk to, and I can tell you that he's going to be talking about cool things.
And that I like spicy food.
And he likes spicy food and can tolerate even when it's too spicy. So without further ado, we are going to hear about, and then this is the other one, is I say phage therapy, pharmacology, ecology, and complications of spatial structure. It could be, what's the Phage?
No, I don't go there.
Yeah, you don't. Take it away.
All right, so I'm at Ohio State. I'm actually at a regional campus of Ohio State. So that's why I'm an hour away instead of 2 hours away. But my tenure initiating unit, as it's called, my TIU, is in Columbus. So I'm a member of the microbiology department there. And so, yeah, I'm going to talk about a lot of stuff and hopefully do it in a reasonable amount of time. So I first gave the talk that looked like this, and it might have been in Zurich. So I gave 3 talks, which we'll get to over the past summer. One was in Zurich, one was in Yanji, China, and one was in Hiroshima, Japan. And the big joke is that I point to this and I say, that's not Ohio. But you guys kind of know that. But that's Sedona, Arizona, and I split my time. I'm about, I don't know, a quarter of the year in Sedona and about 3/4 of the year in Ohio. And my wife lives there and my son lives there, and I live part-time in Ohio with my daughter who's attending Ohio State right now. But anyway, that's why that's there. And then this I put up because before I gave the talk in Yanji, China, it was the opening talk. I was told that I should kind of give them some sense of who I am, because they have no idea. And so I said, well, maybe I should show them my books. That'll give them a sense. So I put this together with my books. And then there's this unfortunate thing that I've got 3 more books in the works. One of them is sort of done at this point, but they have become the bane of my existence. I have declared that after this, I am not doing any more books, except for writing my own books. It's the edited volumes that are just killing me. Ah, yes. So these are the talks I gave last summer. So this is the Yanji, China talk, Considering Phage Therapy Pharmacology, the Pulmonary Perspective. And this is the Hiroshima talk, Applied Ecology of Phage-Mediated. Sorry. That's okay. Otherwise it would be boring. And this is the Zurich talk, Ecology of Phage Biofilm Interactions, Theory and Application. So basically I gave 3 completely different talks. And then I gave a talk in Vienna, and I decided the easiest way to do it was to combine all 3 talks. And it occurred to me that I could use these talks, or their titles, as a way of kind of guiding you through the talks themselves. So the first part that I'll be going through as I try to cover a bit of ground is just an introduction to phages. And so phages are the viruses of bacteria. We know these guys, especially with their spider-like tail fibers, as being the typical, not just phage, but virus generally. When people think of viruses, they think of phage T4. But there are tailed phages. There are phages that have lipids associated with their, I guess, capsids. And then there are the single-stranded phages. So there's a fair diversity of phages. Interestingly, the archaeal viruses are as diverse, if not more diverse, in terms of their morphological structure. And this despite the fact that we know about 10,000 phages, but only about 100 archaeal viruses. So it's a lot of diversity among the archaeal viruses. And this I put together in part because I love making overly complex things, or taking simple things and making them complex. Although this is a complex thing that I made complex. I just tried to put everything I knew about phage infections into a single image or figure. And I was able to do it because I wrote a book. I like writing books, it's editing books. And I wanted to put figures in it. So I published this in my book as a figure, basically an introduction to the complexity of phage infections. And that there I circled is basically the lytic cycle, where you have adsorption and then you have the process of making new phages and then lysis of the cell. And then I looked at that and I said to myself, that looks a lot like the yellow submarine of Beatles fame. And so that's the yellow submarine. And so then I kind of tried to see whether or not it really was the yellow submarine. So I tried to fit the data to the theory, and it didn't work out very well. So I decided instead to fit the theory to the data, and that seems to work just fine. So anyway, so one of the things I do is I like historical things. I like old papers a lot. Kind of like Ledsky with his love of old books. Although I like to scan them. I don't want the hard copy itself. I want it on my computer and I want it OCR. But one of the things that I did, I don't know, it was 2.5 years ago, is I decided that there was many different definitions of secondary infection as far as phages go. I mean, you can see examples of this here. It has something to do with superinfection immunity, where you're blocking the ability of a temperate phage to replicate an already temperate phage-infected cell. Superinfection exclusion, where you're preventing a second phage from actually getting into the cell. Something called lysis inhibition, which I've actually studied for 25 years now, or is it longer than that? I'm getting old. But anyway, this idea of superinfection, secondary infection, I decided to explore a little bit more, and I came up with this idea. There's actually 3 different definitions of secondary infection in the phage world, and only 2 of them are important. And all 3 of them are also found essentially in medical microbiology. And I named these parallel secondary infection or superinfection, serial secondary infection, or this mixed parallel serial. And parallel basically means you have more than one infection going on at the same time. So you have an infected cell, second phage comes along, absorbs the cell, and at least attempts to infect it, and get co-infection. Serial is from epidemiology, where the primary infection or the index infection would be the first infection within a population, and that gives rise to subsequent or secondary infections in the population. So anyway, I do stuff like this, is find interesting words and just turn them into papers. And this one actually was accepted for publication. There it is. And it's sort of in press right now. In Virologica Sinica. I was like, okay, they're going to do a 100th anniversary of Phage issue, and I figured I would submit something, and so I went through all the manuscripts that I hadn't completed, and I found this one. I said, yeah, this would be perfect for them. And it's actually, it's not even in press, it's actually sort of out there. And so one of the things I did for the Zurich talk is I decided nobody knows that I publish a lot, so I thought I'd put images of the first page of all of my publications. And this is not all of them. This is the one that consider phage biology, ecology, a role in bacterial pathogenesis, and I'm also into phage history. And so this is the new one in Virologica Sinica. So then we move along. So phage therapy is something I spend a lot of time considering, thinking about, writing about. And phage therapy is the use of bacterial viruses to attack bacteria that we don't like. And the narrow definition is that we use these phages to attack bacteria that are actually causing infections. A broader definition of phage therapy is that we use it as a means of antibacterial biocontrol. So this could be in the environment, for example. People have been doing phage therapy continuously. This is within the clinic for 80 or so years. It's actually been around for going on 100 years right now, maybe 95. Phages are natural products. They're abundant, they're diverse, they're diversely bactericidal. There's lots of different ways that they kill bacteria. Antibiotic resistance in bacteria doesn't seem to translate into phage resistance. If you choose the right phage, they're not toxic either to our bodies or really to our microbiomes. If you choose the wrong phage, however, for example, the phage that's associated with the production of cholera toxin or Shiga toxin or diphtheria toxin, that's not a good thing, but it's possible to choose the right phage. You have very low toxicity. They can display the favorable pharmacokinetics of actually amplifying within an infection, in the course of treating an infection. So they increase their numbers while they're attacking the bacteria that they infect, which makes sense because that's what viruses do. They increase their numbers while attacking the cells that they infect. The formulations of cells can have significant shelf stability. They can last for months or years. And there are a number of companies that are in fact involved these days in developing phage therapy. And this is a list of companies. This is actually probably from June or July, but this is a list that I maintain on the ISVM, or International Society for Viruses and Microorganisms website. It's just a bunch of phage companies. This is something I put together last June. It is the number of phage therapy experimental papers that have come out per year. And we're only up into the range of 25 or 30 per year, at least in English language publications. But you can see that there's a steady increase. And there seems to have been like dozens that have come out already this year. I'll get to that, but it seems to be a growing field. Again, it's this use of the viruses of bacteria to attack bacteria that are nuisance or pathogenic. This is actually, at least as of last September or so, the complete listing of experimental papers using phages to attack bacterial pathogens of plants. It was a plant pathology meeting that I was at in Hiroshima, so that's why I put that together. And I also have begun blogging, and so this is actually something that I've put onto this, a smaller flea blog that I'm a part of. And then this is a website that I try to maintain. Maintaining websites is an impossible dream because they're always in need of updating, and if you have too many of them, it just gets overwhelming. But this is one that I have called Phage-Therapy.org, where I'm trying to basically present all that's important in phage therapy to the world. And then moving along, the pulmonary perspective. This is actually based on a manuscript that I submitted or resubmitted yesterday. And so there are actually not that many phage therapy studies for the treatment of lung-associated infections, although I submitted this in November, and since then something like 4 or 5 new studies have come out, so it seems to be going someplace. But this is just a number of different studies, and the interesting thing about them is that the use of phages against these lung-associated infections seems to be effective. I mean, a lot of them they're getting 100% efficacy, some of them they're getting only like 77, 87% efficacy, but that's pretty good in treating infections with these viruses. This is out of a 1987 paper where they basically had, I guess, about 100 or 150 different cases they were talking about of treatment of pulmonary infections. There were a lot more cases they talk about or summarize. And what we have here is that these are the cases that the phage therapy was actually successful for. So it's in the range of about 80% of the treatments that are actually successful. And this is particularly interesting because generally, this is done in Poland. Generally what they're treating are infections that are chronic and resistant to antibiotics. So basically these are people that can't get rid of their infections, and they come to these phage therapists. They now have a phage therapy center there. And their infections are treated and actually cured, even though they're these chronic infections that otherwise won't go away. And this is an experimental paper in mice. What they did is they immunocompromised the mice, and then they infected the mice with Burkholderia, and then they treated the mice 24 hours after the bacterial challenge with an aerosol of phages, which is impressive, getting an aerosol into mice, and also impressive that they waited 24 hours. And this bar here is showing a 2-log drop, and so this is actually the number of phages given treatment, and that's, number of bacteria given treatment, and that's the number of bacteria without treatment. And here out by, is it 7 days? Something like that. Something like that. They have substantially fewer bacteria. So it's a nice little model they put together. And the phages are really wiping out these bacteria, even though they've allowed the bacteria to become established to some degree. There's a paper that just came out, or is about to just come out, where they actually encased phages in liposomes and injected them into the blood of, I believe it was mice with a Klebsiella pneumonia model. The phages inside of these liposomes were actually able to find their way into the lungs and substantially reduce the bacterial load in the lungs. So it seems to work. And so I have an interest in phage therapy pharmacology, which I don't think I had when we met last. This is something only since like 2009 or 2010 that I've been doing. I came to the realization that if you're going to be interested in the ecology of something that people use as a living drug, then that's almost equivalent to the pharmacology of this thing as a drug, and I have been doing this ever since. It helps that my wife has a little bit of a pharmacology or a pharmacy background, so I was a little bit primed to do this. And so what I've been doing is getting involved in trying to understand phages as drugs, or from the perspective of them being drugs, their pharmacology. This is just showing that the place of phages among the grand scheme of antimicrobial agents. So you have antimicrobials, antibacterials are a subset, disinfectants a subset of that, antiseptics a subset of that. Then you have the antibacterials that display truly selective toxicity, and among them are the phages. So with the phages, there is a flexibility in how you apply them. You can apply phages topically, you can apply phages directly to the blood, or in a way that they easily get into the blood parenterally. Paros refers to oral taking of drugs that are then taken up by the body. If you apply phages topically, they have much more direct access to the bacteria that you're treating, and this has the potential to allow you to use greater phage numbers. If you're doing less direct application of the phages, then you have a need for the phages to, in fact, replicate once they reach their targets. With drugs, what you do is just increase the concentration until it's high enough at the targets. With phages, there's limits to how high you can increase the concentration. So people are really relying upon these phages to find their way to these infections, great densities of bacteria, that actually start infecting the bacteria there and making more phages, which then go on to wipe out the bacteria. And so this is another way of looking at the pharmacology of phage therapy. So what we have here is you apply the drug or the phage, it increases in number, and then there are mechanisms that decrease the number, the density of the drug or the phages. Here we have basically the concentration in the vicinity of whatever it is the target is. And then here we have the minimum effective concentration and the minimum toxic concentration. So your goal when you're applying drugs, treating with drugs, is to keep the drugs within a range that's above the minimum required to be effective, but below the level that is actually toxic. With phages, there are two important things that allow them to be, in some ways, more effective than other antimicrobial drugs. And the first is that they're not very toxic. So that means that you can apply phages in high concentrations, or since the phages are actually replicating inside of bodies, inside of wherever the infection is, you don't have to worry about them increasing their concentration substantially in the vicinity of the infection. And the second thing is that, of course, phages are viruses, so they can replicate. So this I consider to be an aspect of metabolism. Absorption, distribution, metabolism, and excretion are all pharmacokinetic terms. Absorption is getting into the blood, distribution is getting around to the rest of the tissues of the body, metabolism is modification chemically, and excretion is removal from the body more or less intact. So I consider metabolism for phages to describe their ability to increase in number by infecting bacteria. And so here we have two different ways in which, two different philosophies in a sense of how phage therapy can be, I guess, practiced. One of them is called passive treatment, the other is active treatment. Passive is also called inundative. And the idea there is you're using phages just like a drug. You have to supply enough phages so that the phages that you supply are sufficient to have whatever effect you need. So in the case of an antibacterial drug, it would be eliminating the bacteria. And this is inundative treatment because you need to supply enough phages to wipe out all the bacteria. Whereas active treatment is this magical thing where the phages go in, they infect bacteria, they make more phages, and that has the effect of increasing the local concentration of phages to the point where you can wipe out the bacteria. Now the thing about active treatment is that it requires higher phage performance. So the phages, they have to not only attach to the bacteria, but they have to successfully affect lice and amplify their numbers, which is not necessarily something that every phage is going to be able to do against every bacterium in every infection. Whereas just having the phages absorb and kill the bacteria requires much less of the phages than having them actually grow. And in addition, you need to have a sufficient number of bacteria around to support phage population growth to high enough densities that you can wipe out the bacteria. So active treatment is harder than passive treatment. This is something that I put in to try to kind of synthesize these ideas. Down here we have the phage density at the point of application. And here we have how much the phages have to actually increase in number in situ in order to have sufficient bacteria killing. And over here, corresponding to this, we have the actual number of bacteria. If you have enough bacteria around and you actually have the right phages, the phages themselves that are present naturally might wipe out the bacteria and actually serve as a protection. There are people who are thinking about this idea that phages may actually contribute to our ability to resist bacterial infections. In here, we have this active treatment where you've got lots of bacteria around. You can supply phages. You don't necessarily have to supply a lot of phages, but if they can replicate enough, they can reach high enough densities to wipe out your bacteria. And then at this end, you've got this passive treatment. Even if you don't have a lot of bacteria around, you can still have phage therapy. You just have to supply all the phages you need to begin with. And then you have in between this idea of passive-active, where the phages are supplied in high numbers, but the replication of the phages might actually contribute to the phage numbers in such a way that it increases the ability of phages to wipe out the infection. Now, one of the things that's missing from here is that it's also can be a good idea to repeatedly dose with the phages. This is what you see in the clinical treatment of phages, is that people will repeat the dosing over and over, sometimes for even weeks or months to get rid of these chronic infections. This is not something that you see a lot in the experimental animal literature for phage therapy. So this idea of needing enough phage and needing enough bacteria around in order to allow the phages to grow to inundated densities is essentially equivalent to the idea from phage ecology of kill the winner. The idea that you can have, especially in aquatic systems, a high diversity, for example, of cyanobacteria, because those cyanobacteria that are particularly successful grow to high enough densities that they support phage population growth to essentially inundated densities and wipe out the bacteria. So this idea of active treatment is not that different from the idea of killing the winner, where you have to have essentially winner bacterial densities of the pathogen in order to wipe them out, which kind of happens when you have an infection because you have a monoculture, so you in fact do have high densities of a given bacterial type. And then over here, phage inundation threshold is something that people, a term that's not used that much, but it's used to describe the number of phages that you need to add to a system just in order to essentially get rid of the bacteria. We'll get back to that idea. And then these are my phage and phage therapy pharmacology, phage therapy and phage therapy pharmacology papers. And this is the one that I just resubmitted yesterday on pulmonary phage therapy that I'm hopeful will be accepted, so I don't have to worry about it too much more. Then we have the ecology of phages. So this is sort of where I began. My first paper was published in 1989. And what I do is what I call evolutionary ecology of phages. I'm interested in the evolution of their adaptations, their phenotypes. And for the talk, I think in Hiroshima, I decided to pull together a list of the most basic issues to phage ecology. And one of the issues is what is the density of bacteria, which we've already talked about a little bit. You need enough bacteria around in order to support phage population growth. So it's an important determinant of the rate at which virions find bacteria, the density of the bacteria. It actually can contribute to determining the phage generation time. The longer they take to find the bacteria, the longer their generation time. And what I call effective burst size. The longer it takes them to find the bacterium, the more likely the phage is going to die, become inactivated in the course of that. And therefore, the fewer phage virions that are in fact going to survive until the next generation. Burst size is a description of the number of phage virions produced per infection. It's an important determinant, as it makes sense it would be, since it determines generation time in part of how fast the phage population can grow. And then in terms of phage therapy, if you have low densities of bacteria, you're going to have to use a passive treatment. If you have higher densities or high densities, you can use this more active treatment. And then there's something I call active penetration, which we'll also get to, which is using phages to treat biofilms. I have a whole book on basically phages and biofilms and the treatment of biofilm bacteria via phage therapy or phage-mediated biocontrol. And the active penetration is the idea that these phages may be able to penetrate into biofilms by essentially lysing the bacteria one layer at a time. And that takes the highest level or highest concentration of bacteria. And then there's also phage density. So there's a transition ecologically going from lower to higher ratios of phages to bacteria. That's what the secondary infection thing that I spoke about is all about, where you suddenly end up with certain kinds of phenotypes, such as a higher likelihood of lysogenic infections when you have higher numbers of phages versus lower. In terms of applied ecology, you only get substantial killing of bacteria if you have high numbers of phages around. And the numbers of, and I need to write a paper on, my argument is that high basically means about 10 to the 8 phages per ml. And then the numbers of phages within a system that you're treating with phages for phage therapy is going to be the number that you add plus the number that you generate via amplification minus any losses due to either dilution or inactivation. And then we have something called phage, that I call phage performance. So this is a paper, from a paper I put together where I was taking all of the bacterial resistance mechanisms to phages and try to figure out what kind of analogs exist in terms of especially animal immune systems. And although I'm not going to go through all of this, the paper was, I guess, published in 2012, what all of these things do is they are a bacteria impact on the performance of the phages. So what they can do, the bacterial adaptations can do, is degrade phage performance in terms of how fast the phages can find and attach to the bacteria, in terms of the burst size, the number of phages made per infection, and also in terms of the generation time of the phage. And so the phage performance can be degraded by mechanisms that are bacteria encoded. And then the last thing is phage penetration. The phages have to actually get to the bacteria. It's easy to think about what goes on in broth cultures that are well mixed, but when you start considering spatial structure, it's not necessarily going to be that easy for one organism to find another organism. And this is basically the ability of phages to reach bacteria, which is something we'll talk about also again. And then there's everything else. I put this together for a paper, I guess it was published in 2000, or a chapter, I guess it's a paper, published in 2011. I just tried to come up with every interaction I could possibly think of. I tried to write about at least some of these, but it's a little bit complicated. So I'm not going to try to go over it right now. And so these are my lysis, latent period evolution, and phage population growth papers. It's kind of where I started. So the applied ecology of phage-mediated biocontrol is the title of the Hiroshima talk. So this again is that same figure showing all of the ecological interactions. And I updated the figure. I changed communication or interact or whatever into interaction. And then I went through it and I tried to figure out everything that wasn't relevant to phage therapy, and I graded out. And I was going to talk about this at the Hiroshima talk, but I decided there's absolutely no way a person could talk about something that complicated and long. So I never quite got to it. But I did put together a manuscript called Harnessing the Applied Ecology of Phage Therapy. And one of the reviewers said basically, why did you even bother to write this manuscript? And after I read that, I was like, well, I don't have enough time to actually bother to reply to reviewers' comments. So it's kind of sitting on my desk right now. But I thought that maybe a better title for it would be What's Good for the Phage Therapy Isn't Necessarily Good for the Phage. And an example of that is when you're using phages to treat biofilm. So I'm thinking of the microcolony of bacteria as being a basically unit of a biofilm. And there are a number of different ways in which you can remove a biofilm, thinking about them as these microcolonies. One of them is you can mechanically remove them, either scrub them off, or you can have, for example, animals that scrub or scrape them off of surfaces. You can actually dissolve the extracellular polymeric substance that glues them all together. In fact, there are phage enzymes that do that. And you can purify those enzymes and use those to release the bacteria from the biofilm. And that might be good for removing the biofilm, but it's not necessarily good for the phages because it's having the effect of actually potentially reducing the concentration of phages and the concentration of the bacteria. And the lower the concentration of the bacteria, the longer it's going to take for the phages to find the bacteria. We have these proteins called indolysins, which can be used to lyse bacteria from without, and that's a way that you can get rid of biofilms. And then you can simply add phages, and the phages can find their way into the microcolonies and actually replicate and kill off the bacteria. This is what the phages normally do. All these other mechanisms are not necessarily good for the phage, but that's how the phages normally would attempt to exploit a biofilm. But as we'll see, they're not necessarily highly effective at exploiting biofilms in nature. Which leads to the ecology of phage-biofilm interactions. And so this is spatial structure. And so these are lots of different ways you can think of spatial structure in terms of bacteria, or for that matter, phages. And so just concentrating for a second on microcolony. So a microcolony essentially is what it says it is. It's a colony that's very small. It's grown up from just a single cell. And in fact, I got started on all this spatial structure stuff, at least in part, because I looked through a dissecting scope at a phage plaque. And there within the outside, the periphery of this plaque, the cloudy part, if you look at it macroscopically, were all these little dots that I knew were basically microcolonies that the phages hadn't completely penetrated into and lysed yet. And so it got me very interested in phages and interacting with microcolonies. This is my idea of the life cycle. Well, it's not my idea, but it's my representation of the life cycle of a microcolony. If you think of microcolonies as being, in a sense, organisms unto themselves, then the job of a microcolony is to make new microcolonies. But at steady state, for every microcolony that exists, on average, it's only going to produce one new microcolony over its lifetime. So this is the idea that this microcolony might be producing lots of disseminating cells. Most of those disseminating cells die. They never find a surface to land on and form a new microcolony, and so on. So just a very simple idea. Of course, if they're increasing in number, then it's not going to be a steady state. If they're decreasing in number, it is a steady state. And so I applied that idea to phages and their interaction with biofilms. And particularly I'm talking about not so much phage plaques as they form on plates, but instead what I call a focus of infection, which would be the equivalent of the phage local exploitation of a biofilm. It finds a biofilm, it starts making more phages which spread and are able to kill other bacteria that are locally present. And so this is the same idea. Instead of using microcolonies, we're talking about phages, where a phage finds a biofilm, starts making phages, lots of phages, but at steady state, no matter how many phages are produced, on average, only one phage is actually going to find its way to another biofilm to infect, to eliminate, to produce another focus of infection. If you think of, for example, a pond, I mean, there's a lot of non-surfaces in there. There's a lot of biofilm, but it's not highly concentrated in the environment. Lots of different kinds of bacteria. Phages are very specific for the kinds of bacteria they infect. And so as a consequence, the phages are not living an easy life. And so they have to produce lots and lots of phages every time they find a biofilm, multiple infections if they're lucky, in order to produce enough of these phages that go off to diffuse away to actually find their way to a new biofilm to initiate another focus of infection. Again, if the phage density is increasing, it's not a steady state, so you're getting on average more than one phage finding a new focus. If it's decreasing in density, it'll be on average less than one. But at steady state, you would expect no matter how many phages phage can produce per focus of infection, on average only one is going to initiate a new infection, or a new focus of infection. And that leads to this. So this is my model for what goes on in terms of phage exploitation of biofilms from the perspective of biofilms consisting of microcolonies of clonal bacteria. So you have a phage that comes in and it's able to infect a cell, making up these microcolonies. That cell lyses, produces more phages. Those phages can infect other cells in the same microcolony, or they can diffuse to close by microcolonies within the biofilm, or perhaps further away microcolonies within the biofilm, producing within this focus of infection phage virions, and some of those phage virions, maybe quite a few of them, are able to diffuse out of the focus of infection, and those are the ones that are charged with finding new places to absorb, new biofilms to absorb and initiate new foci of infection. And the argument is, in fact, at steady state, no matter how many phages are being produced in this focus of infection, on average only one is going to find its way to another biofilm to initiate a new focus of infection. It's a hard life for these phages. The phages that are exploiting biofilms don't seem to be very numerous, but that doesn't mean they're not around. And their job, of course, is to make new phages. And so I'm just pointing out these things I call explorer phages. That's the equivalent of that arrow right there. And I guess this is my spatial structure plaques and biofilm phage studies. And so, summing things up, we've got passive treatment that requires attachment, infection, and then some kind of bacterial cytal activity. Nothing more by definition, because you don't require phage population growth to wipe out the bacteria. Active treatment, you actually have to have absorption, infection, bactericidal activity, lysis, and amplification. Somewhere in between is this idea of passive active, where you've got enough phages around to wipe them out with absorption alone, but this might be improved in terms of its effectiveness by the fact that phages can replicate. And it's my feeling that this serves as some kind of an ideal for phage treatment, that you want to have enough phages around, whether you've applied them or whether they grow globally within a population, within a community of bacteria, in order to wipe out the bacteria, but it's awfully nice that the phages are able to replicate while they're there and increase their numbers locally even further. And then this is active penetration, where you have absorption, infection, bactericidal activity, and lysis, which may in some manner increase the penetratability of the bacteria that are found deeper within these biofilms so that the phages can wipe them out. And as I like to say, so phage behavior during treatment of biofilms may take on something like that. And as I like to say, applied ecologically, you have to dose again, because you just got to keep coming with the phage and hitting with them, hitting the bacteria with them, hitting the bacteria with them, in order to get rid of these chronic infections. Because probably the phages don't grow that well, don't replicate that well when they're infecting these chronic old bacterial infections. And then there's this, so I almost don't do anything, it seems, towards doing any of that work of late, because all I'm doing is teaching. And this is the center of my teaching, and this is my website. And right now I've got actually over 250,000 users per year and 500,000 page views per year. And on there I've actually got 2 online books. One is kind of a strange book that has a lot of YouTube videos in it, and the other one is my attempt at talking about at least microbial evolutionary ecology at a somewhat basic level, although it's unfortunate because I know that there are a lot of people in this world who understand microbes and evolution better than I do, but I've got a book that's online. And that's it.